Hi, folks. My name's Patty. We're going to wait just a couple minutes for some more folks to show up. So if you could stand by for just a minute, we'll be starting up shortly. All right. Well, it's 6:01. We may have some folks wandering in the next few minutes, but I can at least get us started here. My name is Patty Casey. I'm the Environmental Surveillance Program Director at the Agency of Agriculture. And a part of my job is to work with Brooke a little bit, and I've been learning a whole lot about apiaries. So I'm really excited about tonight's webinar. I'm going to be learning along with everybody else. So a couple of housekeeping items. I would ask everyone to please mute your microphones. And let's see, this meeting is being recorded because we would like to be able to share this with everyone else. We're going to post this to YouTube afterwards. So you are being recorded. Or well, you're not. We are. Let's see what else. So the way that we're hoping to have this webinar go is that Brooke is going to do a presentation. Oh, I think somebody's mic might be on. So if you would just check your mics and turn them off. Brooke is going to do a presentation that's going to be maybe about 45 minutes, and then we're going to have time for a discussion afterwards. So we're hoping that we can hold discussion points until the end. If you have a question, sort of an immediate or urgent question about clarifying a point, you can use the Q&A button, which is should be up at the top of your screen. It just says Q&A, and you can put a question in there, and I'm going to be monitoring those. I guess alternatively, you could raise your hand, although that may be a little harder for us to keep track of, but we will get to you regardless. Let's see, what else do we have here? I think that might be it. So without any further babbling on my part, I would like to introduce Brooke Decker. I'm sure many of you know Brooke. Brooke has served as the Vermont Agency of Agriculture's pollinator health specialist and apiculturist since 2019. She's currently the president of the Apiary Inspectors of America. She holds a Master's of Science degree in Environmental Science from Antioch University, and she's an absolute joy to work with. So I just want to add that little side note. So Brooke, thank you, and take it away.
Thank you, Patty, for the kind introduction. So I am going to start sharing my presentation. So looks like it's loaded on my end. Do you see that on your end?
We do. That looks great.
Excellent. All right. Well, I don't need to introduce myself too much because Patty gave me an introduction, but this is a Tropilaelaps webinar. So the first of maybe many Tropilaelaps webinars. So welcome, and I hope you enjoy the show. So to start with, this is the most challenging part for some of us, is saying the word Tropilaelaps. So it is spelled out Tropilaelaps, but it's very easy to just say Tropi or Tropi mite for short. Apparently I have some animation here. I wasn't sure about that. But so for the remainder of the presentation, I'm going to probably use terms interchangeably, but when I talk about the Tropilaelaps mite or the Tropi mite, I am talking about the Tropi mercedes species and its association with Apis mellifera or the honeybee. So just that association, just for clarity on this subject.
Hey, Brooke, sorry to
Yes.
interrupt. We have someone's hand up, so I'm assuming there's some urgent question, like maybe
Okay,
people aren't
yes, go
seeing.
ahead.
So it looks like Donald, you have your hand up. If you want to unmute yourself and let us know what is going on for you, that would be good. No display. Yeah. Is anyone else having trouble seeing? If somebody, you could raise your hands if you're not seeing the display.
Okay.
Someone else says the same. I can see fine. Okay. Huh. I can see
so
it.
some
All right.
people can see it.
You can see it. So it sounds like it may be on the user's end.
I wonder if it's like a Teams setting, because Teams can be a little different than Zoom's.
Yeah, mostly what I'm seeing is people are saying they can see it, but a few people, yep, Susan says hang up and dial back in. So if you want to do
that helps. That's great.
Yeah.
And I can unshare and then share again. Maybe we'll do both.
Okay.
Does that
Why
sound
don't we
good?
do that? So what we'll do is we'll pause for a moment. Deal. Good. So anyone who can't see, go out and get back in, and we'll have Brooke reshare as well. And we'll try this again.
Great.
Sorry, folks. I'm not sure what's going on, but so it looks like we have 48 people. So it looks like everyone perhaps has made it back in. Let's see. Q&A says looks good to me. Okay, thank you, Ashley. I can see it fine. Okay. Up and dial back in. Okay, try this again. Okay, so we've got people, browser option, try to use the app. Having on their phones. Thank you for all these suggestions, everybody. Really, it takes a village. Really appreciate it.
Okay, I'm gonna present again and see if that makes any difference.
And if somebody, if you want to put in the chat how it's working for you, I will be monitoring that. So let's give it a go here. Opened it in the app. Okay, good. It's back. Very good. Looks like we might be in business.
Excellent.
Works fine. Okay. I think we have at least we have a quorum. Looks like people are writing in that it's working. So, okay. Let's give this a go. I think, go ahead. Different computer. Okay. Thank you, Anthony. Hey, Dave, thank you. Seeing people I know. Okay. Why don't you go ahead, Brooke, and we'll do our best from here.
All right. So in addition to learning how Teams works, we hope by the end of this presentation that you will, by the end of the presentation, understand why we're talking about this Tropi mite and why it's a pest of concern, that you will know about some of the initiatives that are happening now that primarily the ones I'm involved in, that we are working on to preemptively educate ourselves and plan about this mite. And then understand the beekeeper's role, your important role in being part of the solution in this journey. So a little bit of the presentation outline. I'm going to talk about this Tropilaelaps mite, its current distribution and hosts, some of those educational and research initiatives that are happening. We'll go into the basic life cycle, the biology and behavior. Again, very basic. I found myself wanting to go down a rabbit hole, and then I thought, okay, let's just keep it. There's a lot of info to share, so I'm not going to dive deep into the biology, just so you know. That will be a later presentation. So then we'll talk about the different early detection techniques that are happening now and the essential role that beekeepers play in that. And then we'll provide some additional resources and have time for questions. So as Patty mentioned, there'll be some times for clarifying questions, and I entered these kind of looking into a microscope slides as a natural pause for those clarifying questions. I tend to talk very fast, and especially when I'm excited about something and passionate about something. So I'll try to pause on these kind of white circle slides and give room for a clarifying question. And then if it's a deeper question, we'll wait for those at the end. So jumping right in, the Tropilaelaps mite, the original host was the Apis dorsata. So the giant honeybee that has these single comb nests, and they're open comb. They aren't nesting, cavity nesting bees. So they have single comb, and by nature they're kind of a migratory species. They follow the different honey flows and migrate as part of their natural cycle. And so in about 1961, this Tropilaelaps mite, similar to the Varroa mite, you're going to hear me bring this up a lot. They jumped species to they were able to survive and reproduce on the Apis mellifera, the honeybee that we keep. So just broad similarities again to kind of draw a connection here. The top photo are the single colony, Apis dorsata colonies, and just how similar they kind of look to our commercial beekeeping operations, where these are potential routes for this mite to disperse. So dispersal methods of drifting, robbing, and foraging, just to kind of get our brain thinking about how the mite might move. This is a map of the current and the current and past, I guess, Tropi mite. The yellow circle there is a very rough drawn circle of the original distribution, which is similar to the host range for Apis dorsata. So for many years, it was thought that this was a mite that stayed in the tropical area and really was only associated with Apis dorsata, but then it jumped species to Apis mellifera, and then they thought maybe, like, there are studies that talk about how it couldn't move, but as you can see, the arrows in this left corner, that there have been recent reports in Georgia and in that far western region of Russia. So the Tropi mite is on the move, and you'll see South Korea too. So these are areas that are more temperate in climate and outside of that tropical range. So this is the Varroa mite map and its dispersal around the world. So the darker regions, also in the Asian area, to the lighter regions where the US is in the kind of yellow range. But similarly, this Tropi mite is kind of following a similar pattern to this Varroa, the dispersal of the Varroa mite. And so this is why there has been huge concern. So are they really a problem? So we know about Varroa. We just looked at the two maps, and it's helpful to compare the two, but they are not the same. And so I want to reference them a lot, and they're compared a lot, but they're very different. And so we need to learn more about Tropi and not just think of them as the same kind of mite. they're not the same. Their behavior is different. They reproduce faster, and compared to the 1980s, the worldwide movement of goods and things is actually quite more rapid now, I would say. So there definitely, I'm just reading a lot of the words. There's a lot of words on this slide, but the distribution is similar in range to Varroa. The behavior, like I said, reproduces faster than Varroa. There's rapid population growth. They feed on brood under the capping. They can actually inflict multiple feeding wounds on the pupa and cause bee deformities. They cause damage to colonies similar to Varroa, and they spread diseases. They can kill developing pupa. They can definitely wipe out a colony in 1 month left untreated. We learned that when we were in Thailand, which you'll learn more about. The monitoring, which you'll learn more about soon, is they're even harder to detect than Varroa because of their size, and management practices are likely going to be very difficult. So again, this is a photo from our trip to Thailand. How long will a colony survive if left untreated? 1 month. So hopefully in that learning objective that you could agree that this is a pest of concern that we need to be concerned about.
Brooke, we have maybe a quick question.
Sure.
Is Tropi on the move due to human intervention, or is it a natural migration by the bees?
It's hard to know exactly. I think so the region, in my understanding, and I think this may be anecdotal with interviews of beekeepers in that kind of Georgia, Pakistan area, that it could be the borders, I guess, are pretty porous, and it could be from migratory operations, but I think there's researchers trying to determine exactly how they're moving, but likely, I'm guessing, is through beekeepers doing things. And as we learn about you'll learn about the biology, the dispersal, they just don't have a long phoretic phase. So I think it's to be determined, but likely moving bees is probably the most likely culprit. All right, so we'll talk now about some of the education initiatives that we're working on to plan for this mite. So, and part of this was that, I mentioned this slightly, but the researchers looking into this sounded the alarm, and apiary inspectors kind of heard the alarm, and we formed a collaboration with Auburn University and Chiang Mai University and got some funding through Project APIS-M, and we put together this amazing inspector training in Thailand. This was a little over a year ago now. So 8 apiary inspectors, 3 researchers, 2 camera crew, and an industry leader from American Beekeeping Federation. We all went to Thailand together to learn about this mite in its natural habitat and from the beekeepers there. So we studied the biology, we learned about detection practices and management practices. And as apiary inspectors, we were wearing, we always have the hat on of how do we preemptively prepare, and how would we plan for this, and what would we do if the mite, came to the US. So we want to inform ourselves so that we can develop some kind of an action plan and understand what to do if it were to happen. So this is a very busy slide, but I think it really captures our time there. I think it was 10 days there, and I'm not sure I slept at all. It was so busy and full, and we met with beekeepers. We sat through lectures. We helped Auburn on some of their studies that they're doing on different monitoring methods. We looked at tons of bees. We looked at them under the microscope. We just learned so much, and it was an absolutely incredible experience and colorful, as you can see. So after we trained ourselves or got training from the Thailand beekeepers, we came back to the US and set up a training for other apiary inspectors. So kind of a train the trainer situation where we collaborated with a lot more people, and we were able to put together a training just in the past November for 58 apiculture leaders in the US and Canada from 37 different states, provinces, and territories. And this was a big thing to pull apiary inspectors and industry leaders from across North America. And that's the map of participants. So we covered, we're trying to spread the word as far and wide as we can of the importance of being prepared for a potential new pest. So part of this training, was very focused on apiary inspectors, and just so as beekeepers that and understand that we're wearing our hats and trying to do the best we can with knowing the processes that we would have to, work within the government to identify a pest and identify the gaps in the research and maybe regulations by state, and then plan strategically how we would work with it. So we put together flow charts, we had group discussions, and we came up with a lot of great info. There was a lot of great discussions that happened here that at that meeting that, we know this could cause economic impact. We want to learn more about how this mite is spreading. We want to make sure all the stakeholders are at the table when we have these big discussions. So one of the other activities that we did, we had folks look at Tropi mite under a microscope, and then we did this word cloud afterwards. So you may be feeling some of these feelings by the end of this presentation, but I'm hoping that it's maybe like I'm not trying to be doom and gloom. I think in all of our minds we want to be prepared. So we're hoping that there's less doom and gloom and just more understanding of what we need to do to work on this together. So the next steps after the meeting with the apiary inspectors is to educate stakeholder groups, and that's the beekeeper. So here we are, we're doing that. We want to work on an early detection plan where all the stakeholders are involved, learn to identify what gaps might be, what are we missing in order to be prepared for this mite, and then build the knowledge base.
Okay, Brooke, we do have a question.
Okay.
Dave is writing in. I'm not sure it's answerable, but it's a little bit in the direction of what you were just talking about. What's the current ETA in the United States and/or New England?
I think it's a big unknown. We don't know anything, the stuff happens. In the midst of us being prepared for bee pests, the yellow-legged hornet shows up in Georgia, that just like catches everyone off guard. And so, we don't know at all. So I think that's why, we know, we're just trying to educate ourselves. We know it's likely that there could be an incursion into North America at some point, but the crystal ball is broken on that one. Sorry. And so educating stakeholder groups. So this is where we are today. We are spreading some knowledge about the basic life cycle, biology, and behavior of the Tropi mite. So it's a little, we'll just start at the very beginning. Tropi mite is an arthropod, and so an arachnid. So it has 8 legs, and basically when you keep going down to genus, the Tropilaelaps genus is associated with Apis hosts, and so there are, I think my next slide. Yeah, there are 4 species of Tropilaelaps mite. The Tropilaelaps mercedes, as I mentioned, is the pest that we are most concerned with. some papers reference Tropilaelaps that clarier, but we're thinking that there could have been a misidentification because these things are tiny, and they're likely they were talking about Tropilaelaps mercedesae. So there is a reference in some of the old papers that they were a host of Apis mellifera, but there's possibly some debate there. But then there's some other Tropilaelaps mites that aren't associated with Apis mellifera. If we were in Thailand, or other parts of Asia, we would be concerned. But basically, as a reminder, that's just what we're talking about, is this Tropilaelaps mercedesae and its association with Apis mellifera. So the life cycle, again, similar to Varroa, most cells will have one female mite, but could host several. If one leg is laid after 2 days in the cell, and then the mite can lay an egg every 24 hours. So that's faster. I think it's like 1.4 times faster than the Varroa mite. So it can reproduce rapidly. And a total of 1 to 4 mites per cell will happen in the life cycle of the honeybee brood. So again, they're breeding in the honeybee brood and need that life cycle to perform their life cycle. The mature mites mature after 6 days, and the development time to egg to adult to emergence is 9 to 13 days. So again, this is in my mind kind of getting in the weeds, but we don't need to know this quite yet until we start managing it a little bit differently. But 2 to 3 mites will emerge from each brood cell. Basically, it can reproduce faster than Varroa mites, and we don't know a lot about how these bees are reproducing. And again, this is just not to get too into the weeds, but just some image of Tropi life stages and sexes. Is this an animated? Oh, there's an animated slide in here. And then at the bottom, there's some images of male and female. So there's a way to identify them. And the reason to look at this slide is that some of these immature mites might look like a male Varroa mite or an immature Varroa mite. So don't want to get too into the weeds at this, basic presentation. That's how you can identify if they're male or female, is on their some kind of plate. I think they have, not my not my expertise on mite biology terminology, I guess. So a little bit about the behavior that I had mentioned a little bit before. They have a phoretic phase. So phoretic phase, I think a lot of us know about the phoretic phase with Varroa, because Varroa has a significant phoretic phase where they're riding on the adult bees and can feed on adult bees. Tropi may, on the other hand, they may only have a phoretic phase where they're outside of the home or of the brood and potentially on the bees for maybe 1 to 2 days, but they do not feed on adult bees. They appear to lack any morphological adaptations to feed on the bees. So it would be specifically for dispersal, or you'll see some videos later. They're just hyperactive little mites. So it might be intentional. It might be that they're just catching rides because they're running all over the place. Their reproductive phase is less than 14 days. They feed on the hemolymph and fat body. They feed for 2 days when the larvae are at least 3.5 days old. They can use multiple sites on the bee. They prefer drones, but will also feed on workers. And the female Tropi mite does not need to feed prior to laying eggs, and they prefer larvae and pre-pupae. So they basically can reproduce and don't act exactly the same as Varroa.
Brooke, we have a question.
Yep.
Did Apis dorsata co-evolve with Tropi? And if so, how does it deal with it? So it's sounding, to interpret that a little bit, it sounds like wondering if Apis dorsata has evolved mechanisms to protect itself.
Yeah, they have, and we did get some presentations on that, and it's fascinating. If you want to study all the honeybees in the world, go to Thailand. But if I'm remembering correctly, the Apis they did co-evolve with Apis dorsata, and the way they dealt with it, they can shake. So they're exposed combs, and I guess all the bees can kind of shake them off, and so they're very high up in these trees, and I guess the Tropi can just fall down. And then also with their migratory patterns, that brood break, and then these Apis dorsata fly an incredible length. I forget how far, and I think they might make multiple stops. They stop and then will I don't know enough about it I feel like I'm misleading everyone, but I know they migrate, and they can migrate very far, and then they come back and forth. And so I think that was a way that with that brood break they were able to not be infected so much with the Tropi. This is the way they dealt with it evolutionarily. All right, some ID. So some Tropi mite lookalikes. We have a pollen mite that is a lot smaller and maybe rounder. The Tropi mite is there on the bottom. The Barula fly supposedly looks like a lookalike, and then the Varroa mite you can see there. So the real lookalikes, I think I mentioned before, that could get very confusing as we're starting to peel our eyes for any new pests. These male Varroa mites, or immature Varroa mites, female Varroa mites that we might see that don't have that full red coloration yet, could be a potential lookalike. Size-wise, the Tropilaelaps mite is about a third of the size of a Varroa mite, so about the same lengthwise, but widthwise, much narrower than the Varroa mite. And here is a penny in the right-hand corner, to scale, with a Varroa mite and a Tropi mite. So I realize that everyone's screens are a little different, but that just as a size comparison with that penny, you can see how incredibly tiny that Tropi mite is. Again, with the whole penny there, this Tropi mite next to a Varroa, next to a penny. And this is a fun thing that the Massachusetts apiary inspectors realized when they were putting a presentation together, that Tropi mite is about the size of a 0 on a penny. So save your pennies, because they might be out of circulation soon, but that's the size of a mite that we would be looking for a Tropilaelaps mite. So if you have, next time you have a penny, look at that and realize how tiny it actually is. So some visual identification tips. The body is longer than they are broad. The legs are visible from above. They hold their front legs out like antenna, and you can see that in this photo, especially if you zoom in. Do I have a cursor? Can I do that? Can you see my cursor? Oh,
yes, yes, we can. Yep.
Okay, so these here are the front legs that they are holding out like antenna. So when you're doing a hive inspection, you want to look for fast-moving objects. There's a start-stop motion that I have a video for you to see, and there's lots of running on the comb that's visible when you're looking for this mite. You also want to look for spotty brood pattern, deformed wings, mites on the bottom board, and mites that might show up in an alcohol wash or sugar shake. Some of those brood patterns and deformed wings that is associated with Varroa as well. So monitoring. Yep.
Oh, sorry, quick question. What kind of mortality rates or other impacts are being seen coincident with Tropi? And that may be something you're going to address later, but figured I'd throw that out now.
Yeah, so in our collaboration with Auburn, that's the most, I feel like that's the most in-depth conversations I've had with the researchers about the mortality rate. There might be studies. My understanding in Thailand, when we were there, those first images of the beekeepers, they like basically if the bees are left for 1 month without treatment, they will die because of Tropilaelaps. In South Korea, Dan Arel from uh, Auburn, he was mentioning that the Varroa, so there can be Varroa and Tropi in the same hives. In Thailand, the Tropi was dominant, but in South Korea, at least where he was, it appeared that the Varroa were kind of more dominant, and they still had to be managed, but so it was like probably a different mortality associated with Tropi specifically. It seemed to be more heavily, influenced by Varroa, but I'm not sure how he determined that. Maybe that might be part of his next paper or something. I'm not sure. So unknown yet, still. So some of the monitoring techniques that we worked on when we were there, obviously visual inspection of bees. Some of the most promising was uncapping the pupae. We looked at just visual observations on brood comb. We did some sugar shakes, alcohol washes, sticky boards, bump tests, and eDNA. So using some of the same techniques that we know of that are used for Varroa and applying them to Tropi, just that's what we know. So it was, let's see what we can find with these. So some of the visual inspections, these are some photos from our trip. There is a Varroa, or not a Varroa mite, a Tropi mite on the queen's eyeball here. So that was a really interesting find. Again, they don't usually spend much time in the phoretic phase, so that was a shocking find to see a mite on the eyeball there. Here's another mite here next to some uncapped larva or pupa that we pulled out, and Tropi mite came running out. So again, just some visual inspection here. Here is some Varroa and with a Tropi next to it. Tropi on a comb. There is a Tropi in this photo here, and it is tiny. It amazingly challenging to see. They really blend in well with that comb. There's a Tropi there. Oh, this is a video. Okay, let's see this. So you can see here this start and stop kind of running over the combs. That's very much, I guess, like an identifying feature of this Tropi mite, often just circling the outside of the comb. Let's see if we can do another one. Oh, I thought there was another one. Oh, shoot. Well, let's watch it again.
Yeah, I'm seeing people are liking this video and finding it helpful, Brooke. That's good.
Yeah, good. It was, I hear you. I know that on my first time seeing it moving, it really it just kind of sealed an image in my head of what we were looking for. I think I have another video coming up as well. I hope. I thought I put a couple in there, but all right. So some of the damage that we mentioned. So in these, we're in the observations and what to look for, monitoring. the damage looks similar. So when there's a heavy Tropi mite infestation, it looks very similar to a Varroa mite heavy infestation. There's a reduction in brood. There's an irregular brood pattern. You see some dead bees. You see deformed wings. You see bald brood, holes in cappings. The colony might abscond. There's other viruses associated. So it's very hard. It'd be hard to know what exactly is going on until you dug a little deeper. This one's a video. Yeah, okay. So look very, very closely here in the center. And we'll play it again. Hopefully folks can see how tiny. There's a tiny mite running around on the center of the frame, running around the comb. And then we got one more video here of some mites. Look very, very closely. And there are many, many mites running on this frame. Once you kind of focus your eyes to see them, you'll see the movement. So likely heavy infestation on this frame. And then Chris Rosario, the apiary inspector from Guam, was sucking them into a tube and saving them for samples for us to bring home. So just some images of the different monitoring that we worked on while we were there. We were doing helping with the study with Auburn to understand which monitoring methods worked best to see the Tropi, and we'll get to some of those results towards the end. So we did a sugar shake and an alcohol wash and looked at bottom boards and tried to find them on bottom boards. And the Tropi in there, let's see, I think it's here. It's so tiny, you know, and it looks just like a lot of debris that would be found on the bottom of a bottom board. So what was determined in the study was that visual inspection is really difficult. It's not really effective. It's almost impossible when bees are on the frames. Uncapping the pupae and looking at the brood comb is effective, but it's really time consuming. The sugar shake and alcohol wash were not very, I guess they were very limited because there's not many, they don't have the phoretic phase like Varroa do. So monitoring in the way that we monitor Varroa likely isn't going to be the best method for Tropi mite. Sticky board was very effective. We were able to find a lot once we were able to get some magnifying glasses to use. It's very effective and time consuming, but really hard to see the mites. The bump test. So for folks that don't know the bump test, you scrape off brood and you try to knock the larva out. You scrape the cappings off, knock the larva out really hard, and basically sacrifice a lot of brood for the cause. And so then you gather that up and look at the sample under a microscope. And so it's still visual inspection, but it's under the brood cappings where they would be reproducing. And then eDNA is likely the most effective, potentially costly, but few labs offer the service. It's not something that, you know, we could do in our backyards or in our small home offices.
So we have a couple questions. Is this an okay time?
Sure, yeah.
Okay. Let's see. Andrew's asking, do the colonies in South Korea, do you know if they have a broodless period?
I believe they did. I actually can't say for sure. That'd be a great question for Dan, but I know that it was a more of a temperate climate than Thailand, where it was, you know, really there was brood year round. So I believe so. And I think that's why Dan wanted to study there, was because there's some similarities to some of the regions in the US that it could overwinter, but I'm not sure exactly how long a brood break it was. There's more more to come from Dan Arell and Auburn.
Okay, good. I think you answered this. Did you have success finding them with the bump test? It sounded like that wasn't the most successful way to find them. Is that correct?
We did find them. It was it's very kind of sloppy, and it wasn't an easy technique for us. You know, relying on a lot of visual inspection in microscopes. It's kind of the brood kind of turns into a slurry. And there are studies. I think it was there might be the folks at Auburn, and then I'm trying to think. Jeff Pettis maybe has done some studies on effectiveness and what good monitoring techniques might work. I'll talk about this. Oh, maybe the next slide. Yes. So the National Honeybee Survey, I think 40 some states are involved with this. It's a USDA funded National Honeybee Survey. One of the main purposes of this survey is early detection of the Tropi mite. They use the bump technique, and I believe why they use that, it's fast enough. It just like it kind of worked the best for having, you know, different people in the field doing something that can be quick and effective and get enough results that are good enough or something. I'm not really capturing it well, but it was good enough to do some kind of monitoring. They are looking into changing that up, hopefully, to eDNA. There are discussions since these studies have come out with Auburn that that might be a better early detection method. No promises there. It's the federal government, so we're just hoping that, you know, the funding stays flowing to keep this important survey going. The National Agricultural Genotyping Center, which we actually have a contract with, they are a honeybee pathogen lab in Fargo, North Dakota, and as their pathogen panel, so they have, I think it's 15 or 16 different honeybee pathogens now that they will test for if you do the full pathogen panel. And they've added Tropilaelaps, the eDNA, so they use PCR technique. They added that to the panel. So bees that we send in will just automatically get that. So it's a great way. Not every state uses them as a resource, but we're hoping that, you know, that technique of just send, you know, whatever bees are being looked at, like just have some eDNA. And so they've done certain different studies with brood and with bees themselves and high bottom board debris. So they're a great resource and really helping keep the eyes on the pathogens and pests. The apiary inspectors, as I mentioned, we're really working towards educating folks and having beekeepers and anyone looking in hives to be looking out for this pest. And so that's where you fall in as part of the early detection efforts. So the essential role. Do we have any questions right now?
Yes.
Okay.
Let's see. Okay. In the US, who is the lead agency dealing with all knowing about Tropi? Is it Auburn University or one of the USDA labs?
So this is so wait, read me the first. So who's like, what is the question? Sorry.
Yeah, so in the United States, who's the lead agency dealing with Tropi? Auburn or is it a USDA lab? So who's the who's taking the lead?
It's a little, so this is the back a few slides when we were meeting in that meeting in November. This is what we were trying to figure out. So the it's really gets into the weeds of government structure, but basically USDA APHIS would, if it was discovered in, let's just say in our state, we would have to get an official sample taken, and so it would have to be like through an official government avenue. So either like I could come out, or Judy, who is our plant health, she's our SPUD, every federal thing has an acronym. Anyway, we would get an official sample, send it to USDA APHIS, and they would identify it, make sure that that's exactly what it is. And then that would trigger another series of things. So it's not a lead agency per se, but it's many moving parts, and it's like within the government structure, if that helps without going too into the weeds, because we spent a whole day talking about it and understanding roles. So I don't want to bore you all.
I know, yeah, no. And this also ties into that a little bit. I don't know that this is also, there's so many questions around this now, but how have federal cuts played into all of that? I'm assuming they mean federal funding cuts played into all of this. If you know, and are there legislators that we should be reaching out to ensure proper funding and staffing? So yeah, federal cuts, and who should folks reach out to?
I mean, I think reach out to, our delegation. I don't think it hurts at all. It's a moving target. At one point, there were 22 different USDA people that were somehow associated with honeybees let go. So I don't know in what realm and who, there's very not much allowed to be shared. I know some people have come back. The USDA, the lab in Baton Rouge apparently was hit very hard with losses. Folks, I've just heard a lot of folks got let go. So I think it's worth reaching out to delegation and expressing how important, bee researchers, lab folks, all that are to the industry. Hopefully that helps.
Great. So reaching out to our own delegation, to our senators and Congress.
I think so.
Okay. Is there any sense that the bees being bred for hygienic behavior for Varroa will also be able to detect Tropi in the brood?
I think there might be some studies talking about that. There was talk of different breeding projects maybe going on in Thailand when we were there, but that's a little far out of the realm of apiary inspector realm. Like I think there is, but there's something to that, and I can't say for sure what it was, like if it's similar or not. That's
okay. Well,
researcher realm.
Yeah, we can look into that and see if we can track that down. Has dorsata developed tolerance to Tropi?
Yeah, again, I don't know a ton about dorsata, but I think because they've co-evolved, they're able to kind of survive, coexist together better than Apis mellifera. When a new pest comes, it can cause more destruction than something that they've co-evolved with a pest.
Okay. And then these are two similar questions. Are the viruses they carry more mortal to the bees than I'm assuming then they mean than the mites? Are the viruses they carry more mortal to the bees than the mites?
I think that's I don't know if I definitely don't know enough to say, but that might be one of the gaps in research that we just don't know yet. So these are great. There's a lot we don't know yet, or It hasn't been shared too much lately. I don't know.
Okay. And then the last one that we have right now is are Tropi vectoring a lot of the same viruses as Varroa, or are they going to be potential sources of new viruses?
I think there's always potential for new viruses, and there's are some of this same viruses. The deformed wing virus is one. I'm trying to look back in the notes. I had a list here, but definitely some of the same viruses and potentially more. And I think Sam Ramsey in Colorado might be looking into this some. I know he's done a lot of work with pathogens with Varroa. So I think that might be one of the avenues that he's working on.
Okay, I think that's it for, hold on 1 second. Let's see, you said that without treatment, the hive will succumb in 1 month. How is Tropi being treated right now, if at all?
In Thailand, yeah, they have different. I didn't go into that here because I thought, I already had 60 slides. So I thought, well, let's not go into treatments yet because we're just learning about it. There are treatments that they use in Thailand, but there are not any treatments labeled for use in the US yet. So this is where, we're trying to I've been on meetings with where the EPA is present and how to write label laws. And if you want to snooze those meetings, man, they will not keep you awake. But it's important work, and there are discussions happening about how to add a mite, to a label and add, efficacy rates. And there's research going on, so there's a lot to be done there. And I just felt like that was a little advanced for this presentation in particular.
Okay, and then how will we include beekeepers and scientists on our southern border and in South American countries with education, with our education and surveillance?
Yeah, so I can say that Apiary Inspectors of America have recently reached out to the Mexican apiary inspectors, and that was a great building a relationship, and hopefully we're able to keep those talks going. I mean, we work really closely with Canadians and Apiary Inspectors of America, so we're trying to build that relationship and make sure that we're kind of on a united front and understand that we are all working together on this.
All right, I think you're all clear to take off.
Okay, so beekeeper's role. Basically, what we want you to do is be really good observers. As we don't know how long, it will take for the mite to get here. We hope it never does. But when you're looking in your hives, look very closely, inspect the frames, check the sugar shake and alcohol washes really closely, look for anything odd, examine a sticky board. In many of the discussions that we've had as apiary inspectors, we joke that we're gonna just like buy all the sticky boards that we can find, bottom boards that we can find, and just be using those constantly, because I think that might be the best way to constantly be monitoring, because if we have strong colonies with tons of bees, it's going to be really hard to see any tiny little mites. But really just keep an eye out, and hopefully as things progress, we can have more labs online that we can just be sending as many samples as possible to get tested at these eDNA labs. That would be very effective as a early detection protocol. So what do you do if you suspect you find a Tropi mite? First, collect it and store it. Put it in something that it won't dry up, and don't smash it, or try not to make it so that it can't be identified. So you could put it in alcohol, put it in the freezer, and then report it. So reach out to me. You can email agrbs@vermont.gov, call me on the phone, and then it will go through my channel, or I will take it through the channels of government that we have a whole flow chart to follow. So some additional resources, if you want to dig deep into the info here, we have a website called honeybeepest.org. And if you go to that, click on the Tropi, we basically built it out just for Tropi mites, but we want to add more resources. So this is vetted information that is like peer reviewed, good resources for beekeepers and anyone to go. So there's Tropi, there's info there. There is the postcard. Some folks have probably seen it. I have one here. I have some of these, so when we do some live events this summer, I'll bring some. They're postcards where you try to find the Tropi mites. I have some ID cards. So there's a lot of info there. You can print out ID cards, and we have a 2-pager with tons of info. So you can print those out on like on this here. It says looking for more information. You can click there. There's amazing videos from Project APISM that go into the more monitoring methods, more in depth in the research, into the treatments and what people use in Thailand. So way more in depth than I was able to go into here. So we joke that this was our race car of sponsors for all of this research and to bring all this info together to you all. But it really is a great group project, and couldn't have done it without all the support of everyone. And I know you all are probably involved with a lot of these organizations, American Beekeeping Federation, honey producers, and honey board, and things like that. And with that, I want to say thank you. We're 1 minute over, so we did get a little of a late start. So tried to not talk too fast, but fill it all in there for you.
Great, Brooke. Thank you so much. Let's see. Couple questions, and then if people have questions, if you raise your hand, I'll try to keep track of them in order. But for right now, we have a question. Are there approved treatments such as oxalic acid that work on Tropi?
I believe, if I'm remembering correctly, oxalic acid does not work on Tropi. And I am thinking that or it's not very effective, if I'm remembering correctly, it's because that treatment is more for phoretic varroa. I know that formic is better because it can penetrate under the cells. So the mode of action for oxalic, I don't think is as effective for Tropi, if I'm remembering correctly. There is research. I think it was either Rogan or Dan at Auburn have done research on that.
Okay. If a beekeeper suspects a find, who should they call? I think you mentioned they should call you.
Yep,
is there
call
anyone
me.
else?
Yep, and my email is there on the slide that hopefully is still up, and my phone number is there, and yeah, I will.
Great. And let's see, we have a question from Holly. Holly, why don't you go ahead? Unmute yourself and ask your question. Oh, you're muted, Holly.
There you go.
Can you hear me now?
Yep.
Yes.
Yep. Sorry, the chat function wasn't working for me. I was wondering, with varroa mites, sometimes I've heard using the drone frames, they lay more eggs in the drone cells. Is that something that could be effective with the Tropi mite?
I think that's still to be determined. I know that, or from what we saw in Thailand, the beekeepers we worked with, that drone trapping was not a method that they used. I think it's still probably to be determined. So these are some of the gaps in the research that we just don't know. And, you know, I don't know how long it took for us to get to that point with Varroa, but my guess is that it took a few years to understand that, you know, different methods to use. So I just don't, I don't know off the top of my head.
Okay, great. Let's see. Are there any other questions? Go ahead and raise your hand.
I got a
think we
question.
Oh, all right. I don't see your hand come up, but fire away.
I don't know if you can see me, but it's Brian's Vermont Apiaries, Brian Jasinski.
Hi, Brian. How are you? Brian is part of
Good.
our 3036 study.
Oh, very
I
nice. Thank
apologize.
you.
I'm very sorry. I'm making a couple of thousand lip balm, and I just checked my email. I realized, geez, I got to check this out because I want to learn some more. But my question is Formic Pro able to treat this Tropilaelaps mite?
It appears that the formic is, you know, that is a method that could be used. It's not currently labeled according to the EPA for this, but it is. I think there's still, you know, probably trials going on the different strengths that we use in the States versus what, you know, is used in Thailand and other places. So that's probably one of the most promising treatments, I believe.
What was the preferred method over there during your study?
We, you know, the beekeepers we worked with were using more, I might be able to go back, were using more not you know, not formic acid. It was in that slide with all the photos together. It was fluvalinate, I think. They had a bunch of different chemicals. So it's here on the right, you know, right middle screen were the different products that they used. And I'm sure I have it written in my notes somewhere, but there was so much info and translators and whatnot that some of it got you know, diluted by the time I was able to process it. So I think some of the products might not even be legal in the US. So I think as research comes along, we will have, you know, folks know that this is likely coming and that, you know, we need treatments that will be effective. So there are people working on it. I just don't know exactly yet what they might be.
All right. Yeah. I just hope we're all proactive as far as getting something in the United States, just in case it does happen.
Yes, agreed.
That's all I got.
Thanks, Brian.
Thank you.
Thank you, Brian. All right. Holly, your hand is still up. I don't know if you have another question or if your hand got stuck up in the air. Mine does sometimes, but if there are no other questions, thank you all so much for being here, and apologies to those of you who couldn't see the slides, but I did hear from our communications director during the presentation, and he said that he is going to pull together a YouTube link, and we will send that around to everybody that was on the webinar this evening. And it can also be, looks like we have another question here. Ultimately, can both Tropi and Varroa coexist in the same hive? And at what proportion would that be? Would Tropi overtake Varroa?
Yeah, they can, and I think it depends. I think what we were seeing in Thailand is different than South Korea, so there are much smarter people than I am doing the research on that. So I think it depends on maybe location and different management practices and maybe a lot of other factors, but they definitely can coexist in the hives.
All right. Looks like that's it. Thank you all for being here, and we will follow up with a link. And thanks again for being here, and reach out to Brooke with any of your Tropilaelaps or other apiary questions. Thanks so much, everybody.
Thank you. Bye-bye.
Bye.
Thank you.
Thank you.
Thank you.
Thank you.